Hello, everyone. Thank you for joining the webinar today. My name is Adra Bemi, additional. Well, just to say a little bit about myself, my background is in industrial chemistry, and later I completed my master's and PhD in chemical biology at Cardiff University, where I work on enzymes from several organisms, trying mainly to understand how these enzymes couple their motion to the reactions, chemical reactions they catalyze. Later, I worked on methyltransferases using reaction monitoring MS techniques to identify methylated substrates. Also, I engineered RNAs for specific and guided splicing, especially for using high throughput and mRNA sequencing. So I am joined today, as Ajay has said, by Paul, who works here in ChEBI, and their HESP annotation in UniProt. So questions, the webinar on the Q&A. So what do we hope that you achieve by the end of this webinar? The first is that we want you to be able to search UniProt's knowledge base for enzyme data, to be able to explore chemical informations and enzyme kinetics in UniProt's knowledge, understand to download enzyme analysis pipeline. So these are the outlines of today's webinar. We will briefly introduce what the aim of UniProt is and how the UniProt workflow works. After that, we have a quick overview of different ways to search and explore UniProt and UniProt entries. Then we do a deep dive exploring enzyme-specific features in UniProt and some ways to go from known to unknown, especially regarding biocatalysis. And we will also talk about how to download, and then we'll come to the conclusion of today's webinar. As I said, feel free to post your questions in the Q&A during the webinar, or you can wait to the end to ask them. So let me start with this. Imagine a sunny day that you would like to have a chilled drink. And then you got to the freezer and found large blocks of ice. So to enjoy your chilled drink, you will need to probably chisel through this block to get some peace and put them in your drink. But now imagine that at that moment, one of your friends just said to you, "Come, I have some cubes in my freezer." And when you got there, you found that they are already frozen in small cubes. And not only that, they have flavor labels, and you have several flavors to choose from. So imagine how happy you would be at that moment in time. And that is what UniProt is like. UniProt is like a friend with a freezer full of organized ice cubes, each with flavor labels. So at UniProt, we are constantly freezing new ones and adding flavors and labels to the ones we have already frozen. So I will explain what that means in a little while. First of all, I want to explain to you that UniProt is a collaborative effort between Embley ChEBI here in the UK and the Protein Information Resource in Washington, DC and Delaware, and the Swiss Institute of Bioinformatics in Geneva. So UniProt compiles a comprehensive, high quality and publicly accessible resource of protein sequence and functional information. This information includes primary sequences, including sequence of isoforms, physiological protein functions, including reaction, pathways, and interaction, involvement in disease, sequence annotation of domains, post-translational modification, mutations, and then structural data as well, 3D structures, and then ability to use data analysis tools such as BLAST and multiple sequence alignment. UniProt get data from several sources. About 95% of our data comes from the International Nucleotide Sequence Consortium, comprising of ENA here in EBI, and DNA Data Bank in Japan, and NCBI GenBank in the US. We also import several data from Ensemble, from PDB. We also import reactions from Rhea, and our data are manually created by expert biocurators who are experts in different areas. So starting here from the blue side on the left, and The imported data submitted or imported into UniProt are computationally analyzed and grouped. At this point, isoforms of one gene are displayed as individual entries. And this data is then manually reviewed, going from the white ear to which we call tremble, to the gold star reviewed entry that we call sweet spot. Sweet spot entries are the ones that have been manually created and annotated by expert curators, using evidence from scientific literatures to add functional annotations and to the data that were imported initially. And all these proteins are grouped such that all isoforms are grouped together. The reviewed gold star entries are then used as templates to optimize our computational and automatic annotations of the unreviewed trembled entries. So only entries that are manually reviewed have the gold star attached to them. Now I will go through an overview of some of the features in UniProt. Throughout this talk, I will be using lignin found in wood as an example. Lignin is a renewable material made from chemical building blocks, similar to those in crude oil. Although lignin is difficult to break down, some enzymes from fungi and bacteria can catabolize lignin and release several useful products. So I'll be making reference to lignin throughout this talk. So this is the UniProt landing page. There are several ways to probe UniProt knowledge base from here. And we'll go through that. So on this page, you can see from this search bar, you can search by gene name, you can search by protein name, you can search by list of accession IDs if you have them. Or you can also go through the advanced search option to explore, which we are going to explore very soon. So let's say we want to search for lignin. And if I enter lignin in this search bar and I enter and I press search, this is the result you will get at the moment. And from this result, you can see that we have 200,000 plus results. And from here, you can see that only 619 of these have the gold star reviewed and have the gold star. So which means these are reviewed. And the remaining 232,000 have not been reviewed. So if you select a specific, if you narrow down to a specific entry, in this case, Lig B, and it will take you to the entry page itself. And you can see here that you find annotations of the enzyme lignase B, which is unique UniProt accession number, P31838. And you will also find the amino acid sequence, the number of amino acid that this enzyme contains. And as you scroll through this entry, you will find many other informations that will be relevant. You can find the full sequence of the enzyme also at the end of the page if you keep scrolling down. And then at this top bar here, let's just stay at the top bar for now. At this top bar here, there are many informations that you could explore. So here first, we are exploring the entry. And under the entry, we have the function and some other things that we're going to go through later on. But just focusing on this top bar for now, we can, the menu bar, which is the top bar, also allows you to go to the feature viewer. The feature viewer, it's like the variant viewer, is very interactive and contains amalgamations of many functional informations like chain, active site, mutagenesis. As shown here, you can click a dot to show you the details and the publications that are associated with the particular dot you clicked. And automatically as well, the feature viewer has the structural, the 3D view, which we zoom in onto the particular amino acid you click on this feature viewer. You can go through this top as well and find many other things. But something else I want to point your attention to is the external links. So UniProt has more than 180 cross references to other databases. Some of them are valuable for enzymes, and some of them have been highlighted here in red. We can go to Brenda and Sabihake binding database. We also have a cross reference to Campbell and drug bank for enzymes or proteins that are involved and that have active drug against them. Moving on from the search bar, we can also go to the advanced search. So here we'll focus a little bit on the advanced search and tell you some of the things that you could do with the advanced search. At the advanced search here, you can see that we have many features that allow you to be able to restrict your search to a specific field. And you can use Boolean functions like and or not terms to narrow down your search and be specific in the results you're going to get. And you can add or remove these fields as you found necessary. As you can see from here, there is a search field here located at the top that allows you to be able to, that can help you to direct your search to the right category. Or you can go through the list yourself and find enzyme classification, like the EC number. You can find the pH dependence. You can find functions, and you can use all these fields to search for your protein of interest. So you can apply multiple constraints to this search to simplify the results you're going to get. Unlike the search bar where you find everything that contains lignin, you can really narrow this down. And we see an example and very soon. Something else you can do is that you can also do an advanced search for small molecules. So under the advanced search, you can go through and find this small molecule. And when you click on it, you can enter the name of a chemical compound that you're interested in, or you can enter the ChEBI ID. We talk about the ChEBI ID now, or you can enter the INCHI key. And this allows you to be able to find, all the chemicals that are associated with that have all the chemicals and all the entries that have this chemical associated with them. So UniProt uses ChEBI classification. ChEBI is an open access database, an ontology of chemical entries. So ChEBI's ontology classification allows relationship between chemicals and their parents or children to be defined. This enables you to find to query based on chemical class or chemical role. For example, here, because of relationship of UniProt with ChEBI, you can enter the name lignin, and as you can see, as I'm entering the name, it's giving a suggestion. So autocomplete is helping you to find the compound you are looking for. And this lignin will contain all the list of any child that is associated with lignin. So I'll show you on the next slide. On the next slide, you can see here, so going to the ChEBI site just to show this. So a search for lignin would entail all the children under it. And if we have searched for a higher chemical entity where lignin is also a child, the result would include lignin and all children under it. And so this allows you to searching for lignin helps you to search for everything that is associated with lignin. For example, you can search for lipids as well. You can search for glycolipids. So, ChEBI ontologies allow you to be able to search these terms from the UniProt database. So in the advanced search field now, so let's say, for example, we decided to search for lignin under protein name. And then under the function comment, we put lignin. And then we search for lignin under the ChEBI ID 6457 using the Boolean function or. So anything that contains lignin in as a protein name or contains lignin as a function comment, or anything that contains lignin as a chemical ID, as a ChEBI ID, will be brought up in our search. And the advanced search leads us to, or lead us here. You know, as I said before, using the advanced search makes your query to be more specific, and compared to the previous search, which gave 619 results of reviewed entries, this one gave us 432 of reviewed entries. So you can see the search query at the top of the bar here that is listed in the search bar at the top. And there are other examples of search queries that you can use as well. The first one is the one that we just used now, but there are other ones that you could use. You could use EC star. So star is used in UniProt to search for all the terms relating to a specific query. So if you put EC star in the search bar, it will give you every entry that contains EC number. And you can use the Boolean function or Rhea and review entries, and we pull out a lot of things. So you can also go specifically. For example, here we have ECXXX, the fourth line has ECXXX, and the more EC number you put in here, the more specific it's going to be. I will talk a little bit later about EC numbers so that you understand a little bit more how this can be useful for you. So the results that you find is based on how you customized your table, and how your results table looks like is based on your customization. So I have customized mine in a particular way. So here is an example of what we had in the beginning. This is 619 that we searched for lignin, and you can see here that the table contains entry name and protein names and gene names. But if you click on the customization buttons and you choose specific things, you could have a result that look like this. So you could select for entries, and you could allow your table to display mutagenesis, to display the mass of the protein that you are looking at. You can also display the kinetics, the enzyme kinetics, and the associated publication, and those ones that have 3D structure. So all of these can be customized for your search table, so that from the search table you can make informed decision and which of the protein or which of the enzymes you would like to probe further. So now let's take a deep dive into UniProt. In this part, I'll be talking about specific features as they relate to enzymes, and how you can do similarity searches using compounds, and then if you have a protein of interest that you already have, and how to download some data. So we walk through from an EC number to active sites, binding, activity regulation, enzyme kinetics. These are all the features that are available in UniProt, and they can be very, very useful when you are doing the beginning of your search. Like I said in the beginning, if you have a freezer that is already organized and labeled, you could just go there and pull the things you want, and it will make life a lot more easier and simpler. So just to talk a little bit about an enzyme commission number. So the EC number is a standardized numerical classification system assigned by the International Union of Biochemistry and Molecular Biology to identify enzymes based on the specific chemical reactions they catalyze. So it doesn't matter how the structure or the similarity in their amino acid. The most important is the reaction that they catalyze. So the EC number follows the format ABCD, with each digit increasing the specificity. So for example now, so EC111 is aqueous. So based on, EC number is classified into 7 categories. Number 1 is oxidoreductase, up to 7, which is translocases. So class 1, aqueous belongs to class 1, which is an oxidoreductase. But the subclass under it, the 1,1, refers to enzymes that are acting on CHOH groups of donors. And the subclass is now further divided into NAD plus and NADP plus as acceptors. So 111 is an alcohol dehydrogenases that uses NAD plus, and whereas 1112 is those alcohol dehydrogenases that uses NADP plus. So this is how enzyme information number works. And this is also available in UniProt. So when you do a broad search, you can decide to group your results by enzyme class. And when you click on this enzyme class, it takes you to this page where you can see, you can also narrow down to subclasses and filter and find maybe the reactions that you're in, enzymes involved in the kind of reactions you are interested in. In a deeper that I'll just mention that you can also find a filter by annotations in the entry, and the result will only contain entries with that particular annotation. And you just to be aware that you can stack these filters so that you can, for example, if you choose entries with 3D structures and active site, it will give you all the entries containing both of them. So there can be more, but the entries you will find will be the ones that contain 3D structures and active sites. So when you click on that, you can choose any of this entry, and any of the entry you choose, we have some of these features that will be running through now. Number one is that you can find enzymes with active sites that have been annotated, and the residues that interacts with the ligands that bind within the active site. This feature is interactive, so you can click on it and you can see the particular site and where it is located in the amino acid sequence. We also annotate activity regulation. So activators and inhibitors are clearly named, and sometimes enzymes that have biotechnological use will also be annotated to say this enzyme does this enzyme, and we think this is very important for biotechnology, or those ones that have been reported to have biotechnological importance will be noted in the entry. So you can filter for these as well. So, and we also annotate steady state kinetics as well as pH and temperature dependent from manuscripts. So this can be very useful in your purification or assay development. For example, if you're interested in you want to purify an enzyme and you want to check the conditions that will be favorable for the analysis, you can definitely use UniProt to get those basic information to progress with. Reactions are displayed through the reaction, catalytic reactions are displayed here through collaboration with Rhea, showing you the molecules involved in a reaction. So each molecule, as we said before, has a ChEBI ID, and you can probe this molecule through the ChEBI ID, or you can probe them through the Rhea reaction here. So you can search for Rhea reactions on UniProt website. You can search for ChEBI ID on UniProt website as well. So this can be very useful for you to find enzymes involved in a particular catalytic activity. We also annotate cofactors. As a rule, we only include those that are not already listed in the catalytic activity tab. So you can make use of this. You can filter for proteins or enzymes with cofactors. So we also annotate mutagenesis. So mutagenesis feature is very interactive, and it provides summary of the impact of mutations on enzyme activity or stability. So the report in the manuscript will also be reported here, as you can see, to give you information on what variants that might be useful for your experiment. Lastly here, we also annotate post-translational modifications. So for enzymes with post-translational modification, it can be very useful. For example, if if you want to make a recombinant protein of an enzyme, a recombinant enzyme from a different species that have a post-translational modification, it will be useful if you know what the post-translational modification has, so that you know if you need to use inhibitors, you need to use disulfide stabilizers or glycogen to stabilize your protein for your enzyme for purification. So next here, we are going to talk about the similarity search. There are different ways to do similarity search through UniProt. One of them is through a blast search. And we also have what we call UniRef clustering. And then we can also do Rhea compound search. So I will show you an example of all of this. So similarity search using BLAST. For example, you can see BLAST here is highlighted at the top of every UniProt landing page. And also, if you click on the tool under the entry, you will see BLAST here. And this allows you to be able to search the database for proteins that are similar to your search query. So if you click on that and you search for using this the ligase B here, you will get a result like this, and will give you 250 results of proteins or enzymes that are similar, of proteins that are similar to your search query. And you can see out of these, 14 of them have the gold standard reviewed entry, and the others are not reviewed. So that means they have been computationally annotated. You can also do similarity search through UniRef. UniRef is a form of clustering that cluster and UniProt entries based on relationship. So we have 100% clustering, we have 90%, and we have 50%. So if you click on similar protein on the sidebar here, it will take you to this page where it shows you proteins that are similar to your protein of interest, that have been clustered together with your protein of interest. So 100%, those that have 100% identity, those that have 90% identity, which is the one that is clicked here and showing you, and those that have a 50% identity. And with this, you'll be able to find similar proteins that are working in the same thing or a little bit different, depending on how dissimilar you want them to be. So you can go for 50% identity if you want those are a little bit and less and convert like the one we are looking at. We can also do similarity search through compounds or through molecules. And so under the catalytic activity, you can click on view reaction details and to find diverse compounds that have some resemblance to the one we're interested in. So if you click on that, it will show you the reaction that is for your enzyme of interest. And if you click on this double greater than here in front of any of these molecules, you will have a pop-up that looks like this, and the display will give you options to search similar molecules in Rhea, and some other options as well. You can search in ChEBI, you can see the description of the molecule in ChEBI, you can search for similar molecule in Rhea, you can search chemical reactions in Rhea as well. So if you click to search similar molecules in Rhea, you'll be led to this page where you will find the substructure of your molecule of interest. Why is this important? For example, if you're interested in enzymes catalyzing novel reactions and you don't know where to find it, you can probe the database like this. There are other ways that you can also do this. You can do this directly on UniProt website through SMILES, strings, or through Sparkle. But this is not the focus of today's webinar. So you can modify some of these parameters on this on the search to suit your aim. And when we do that search, we have a result here containing 451 molecules. So these are diverse molecules that are related to the one they have just searched with. So this substructure search limits your compound to only those with two hydroxyphenol derivatives. And you can interact with any of these ones to find, UniProt entry containing this molecule. So it might be there as a product or as a reactant, but when you interact with it like this, you will find you can see some of the rare reactions associated with your search, and this can take you back to UniProt to find the proteins or enzymes that have these reactions. So this allows you to find new reactions that might be of interest for biocatalysis purpose, or the ones that are amenable to mutagenesis or mutations, and to accept your substrates of interest. There's another way two options available there. You can use substructure, you can use similarity search. You can do similarity search and reduce the threshold. The threshold is normally set to 0.8, but you can reduce the threshold to 0.5, and this will give you other functionality, other possibility as well. Here, the result returned 423 molecules, but in this case, the compounds given all have a phenolic base, and they're more diverse compared to the one we searched initially because of the threshold reduction, I believe. So here we come to how to download the entries. So if you have, as we've done all this search, if you find the entry you are interested in, you can easily just click on this blue tab here and mark all the entries that you are interested in, and you can immediately download them. You can click on download, and you can download them in different files. So we have faster canonical. You can download XML. You can download them as GFF. You can download them as JSON. So these options are available, and you can download as many data as you want from the UniProt website. And also there is access to programmatically access all the data in UniProt. So we have REST API, we have programmatic access, and you can find them on the UniProt website, how to access UniProt programmatically. You have also had a training previously that explains how you can access UniProt programmatically. So in conclusion, we've gone through some things today. I've talked about the aim of UniProt. We have explained and we've gone through some overview of some features in UniProt, how you can do simple search, how you can do advanced searches, how you can use customization of your tables. We have also talked about specific enzyme features like catalytic activity, cofactors, activity regulations. And we've talked about how you can do similarity search to help you to get to other proteins from what you know to what you don't know. So you might have a reaction of interest, and then you want to see enzymes that are working around this reaction of interest, or that you can use to make particular compound. And how we've also talked about how you can download your data from UniProt. So there is always help available on UniProt website. So if you go to the UniProt website, you will see an email shaped envelope here. If you click on that, you can contact us directly. And if you need any help, there's help available through through documentations and different FAQs that have been answered for you in how to explore and use the UniProt. And you can see them scattered everywhere on the website. So if you can click on that whenever you're on the website and you're struggling with anything, there's always help available. So we have several trainings that are always coming up. So we are always doing trainings and doing webinars. So you can join any of our live webinar training. You can register for more. And like I said, we have done a training previously on programmatic access. So if you want to programmatically access the data in UniProt, you can go to some of our previous trainings and rewatch them. They are available on YouTube, and you can benefit from all this. So with that, I come to the end. I'll just there are so many, like I said, UniProt is a collaborative effort between EBI, where I work, and Pretend Information Resource in Washington and Delaware, and the Swiss Institute of Bioinformatics in Geneva. So the the presentation today also contains some of the works by the Rhea team, and the people working there have been acknowledged here as well. So we are funded by NIHH grants and with support of EMBL and SERI, and then Open Target. So with that, I come to the end of my talk. So I'll hand over to Ajay now.
Great. Thank you very much, Adira, for a wonderful webinar today. And thanks everyone for joining in. Okay, so with that, we can start our Q&A session. So we already had 2 questions and they have been answered already in the Q&A box. But we still have time. So if anyone has any question, please feel free to type them into the Zoom Q&A box. Is there anything worth mentioning, Paul, from the question that you answered in the Q&A box?
Unless the person who asked the question feels like they want us to expand on it, I don't think so.
Okay.
And I think Alan gave an excellent answer, on which we would have struggled to cope with it. Or I would anyway. I've never heard of plastosphere enzyme discovery. I heard about Adura. Is that something you know about?
Sarah, what do you call it?
Plastosphere enzyme discovery.
No.
So it's a really great question from Jackson, who was asking about using UniProt to prioritize candidate enzymes for validation. And I don't think you can really do that directly with UniProt data, but there are some really good AI models and other kinds of models for predicting candidate enzymes for reactions that are trained on UniProt. And you might like to check out these. I put in a link to one of the models. And also there's a resource that's being developed by an environmental research institute based in Zurich. And they have a kind of pathway prediction tool which tries to predict environmental transformations and the enzymes and reactions and pathways. And they also use UniProt to try and link environmental transformations of pollutants to potential genes and genomes. And they developed like a reaction similarity measure that allows you to do that. So Jackson, if you're interested, you should maybe check out those resources and related resources. They might be useful for you. Having said that, if you do have a specific reaction that you're interested in, you can search for that in UniProt and Rhea directly and get candidate enzymes for it if it's one that we've curated. But if it's a transformation that we haven't curated, you'll need to use these kind of predictive methods.